For decades, our main weapon against cancer was, well, it was powerful, but it was really crude. The approach was basically to carpet bomb the body, just hoping we'd kill the enemy before we killed the patient. But over the last 30 years or so, a revolution has been quietly underway. We've gone from using these blunt instruments to deploying molecular smart bombs. So today, we're going to break down that incredible evolution, starting with the biggest challenge of all.
This is the fundamental problem, right? The core challenge of all cancer therapy. See, unlike a bacteria or a virus, a cancer cell isn't some foreign invader. It's a twisted version of us. It's our own cells, just corrupted. So the central question has always been, how in the world do you design a poison that kills only the traitor cells while sparing all the loyal ones?
For a long time, our best answer to that question was traditional chemotherapy. The strategy was actually pretty simple in concept. Find a vulnerability that cancer cells have, and then just exploit it relentlessly. And that vulnerability? Their need for speed.
Chemotherapy's biggest problem is the collateral damage. See, it's designed to kill any cell that divides quickly. But the thing is, cancer cells aren't the only ones that do that. So are the cells in our hair follicles, the lining of our mouth and our gut, our bone marrow. Chemo attacks all of them indiscriminately, which leads to this devastating and very familiar list of side effects.
So how does it actually work on a molecular level? Well, one of the most common types of chemo uses something called alkylating agents. And you could think of these things as molecular staples. They get into a rapidly dividing cell, and they literally staple its DNA strands together, creating what's called a crosslink. The cell's machinery just can't read or copy this damaged DNA, and that damaged signal triggers a self-destruct sequence called apoptosis.
But here's the really tragic part. This approach is a total double-edged sword. The very thing that makes it so effective, damaging DNA, is also its greatest risk. By damaging the DNA in healthy cells, the chemotherapy itself can, in some rare cases, cause a brand new cancer, often a type of leukemia, years after the original treatment is over.
In this, right here, this is the turning point in our whole story. This is where we see the massive shift from an era of blunt force to an era of precision. Scientists basically stopped targeting a general behavior, like just cell division, and they started asking a much smarter question. What makes a cancer cell specifically tick? And could we attack that instead?
Okay, so this new approach led to what we now call targeted therapy. The goal was no longer to just poison all fast-growing cells. The new goal was to find the specific engine driving a particular cancer and then just shut it down.
One of the first huge breakthroughs came in breast cancer. Scientists discovered that a massive portion of these cancers, we're talking about 80%, had a specific protein on their surface called the estrogen receptor, or ER. Now, what this meant was that the cancer cells were actively listening for signals from the body's own estrogen.
This discovery was an absolute game changer. I mean, if estrogen is the fuel, then the estrogen receptor is the gas cap. These cancer cells were literally feeding on estrogen to grow. And this led to a brilliant idea. What if you could just block that gas cap? So, scientists developed Tamoxifen, a drug that basically acts like a fake key. It plugs right into that estrogen receptor, stops the real estrogen from getting in, and effectively starves the cancer. It was one of the very first truly targeted therapies.
Right around the same time, another target was found. It turns out that about a third of breast cancers have way too many copies of a gene called HER2. Now, HER2 is a receptor that tells the cell to grow and divide. So having all those extra copies is just like having a car's accelerator pedal jammed straight to the floor. The cell gets a constant, screaming signal to proliferate, just nonstop.
So what was the solution? It was a drug called trastuzumab. This is an antibody that's specifically designed to find and latch on to the outside of that HER2 receptor. And by physically blocking it, trastuzumab stops the receptor from sending its grow signal into the cell. It's basically like putting a boot on that stuck accelerator pedal.
But if you want the ultimate proof, the absolute poster child for this targeted approach, it came from a totally different cancer, a type of blood cancer called chronic myelogenous leukemia, or CML. And what happened here was, well, it was nothing short of revolutionary.
So scientists discovered that in up to 95% of CML patients, the cancer was being driven by a single freak protein. A genetic accident fuses two genes together, creating this mutant called BCR-ABL. This protein is what's called a tyrosine kinase, which is basically a molecular on switch for cell growth. The problem is, this particular switch is permanently stuck in the on position, just driving the uncontrolled growth of leukemia. It was the cancer's single, identifiable Achilles heel.
So a drug called Imatinib was designed to do just one thing: find and block that faulty BCR-ABL protein. The results were, they were a miracle. Before Imatinib, CML was often a death sentence. After, the 6-year survival rate shot up to this, 88%. Just let that sink in. A single, targeted drug transformed a fatal cancer into a manageable chronic condition.
The incredible success of drugs like Tamoxifen, trastuzumab, and especially Imatinib, it really ushered in a golden age of targeted therapy. It felt like maybe, just maybe, we were on the verge of curing cancer. But cancer is a wily and ancient opponent, and it was about to fight back.
And that is the question that brings us right to the modern frontier of cancer research. We have these incredibly precise, smart bombs, so why is advanced cancer still so unbelievably difficult to treat? Well, the answer, in one word, is evolution.
This data for breast cancer really shows you the scale of the challenge. Our treatments are fantastic for early-stage localized disease. Look at those numbers. But then, look at the dramatic drop for stage 4. That's cancer that has spread. The 5-year survival rate just plummets to under 25%. And that's because metastatic tumors have had more time and more opportunities to evolve ways to resist our best drugs.
See, a tumor isn't a single thing. It's more like a chaotic population of billions of cells, and each one is slightly different. So when we hit it with a drug, we're creating this intense selective pressure. Most of the cells die, yeah, but a few, just by pure chance, might have a mutation that helps them survive. They can develop these tiny pumps to literally spit the drug out, they can beef up their DNA repair kits, or even rewire their own self-destruct circuits. But the most direct escape route? Change the lock. Mutate the very protein the drug is targeting.
Let's look at a really clear example. Remember trastuzumab, the drug that binds to the outside of the HER2 receptor? Well, some of these cancer cells figure out a seriously clever workaround. They evolve a way to produce a shortened or truncated version of the HER2 protein. This new version is missing the entire external part where trastuzumab would bind. But, and this is the key, the internal accelerator part of the protein is still active, so the drug is now completely useless against it.
And so the chess match continues. Our move? Scientists developed a new drug, Imatinib. It's a small molecule, tiny enough to actually get inside the cell and block the HER2 receptor from its internal side. This means it can shut down both the original HER2 and that new, truncated version that outsmarted the first drug. It's a constant back-and-forth arms race between our ingenuity and cancer's evolution.
And to make matters even more complicated, we've discovered another problem, a hidden enemy within the tumor itself. It seems that within many cancers, there's a small population of what we call cancer stem cells. These cells are tough, they're slow-growing, and they are highly resistant to a lot of our therapies. So even if a treatment wipes out 99.9% of a tumor, these few resilient cells can survive and, eventually, cause the entire cancer to regrow. So the journey from chemotherapy's blunt force to the incredible precision of targeted therapy has been truly revolutionary. It's transformed outcomes for millions, but it has also revealed who our true opponent is, not some static disease, but a relentlessly evolving ecosystem. The battle is no longer just about finding the right weapon, it's about winning an arms race against evolution itself. And that leaves us with the ultimate question. In this race, can our ingenuity finally outpace cancer's ancient, relentless drive to survive?